Hello, everyone. We're going to wait for a couple minutes to let everyone to connect, and we are going to start shortly with our presentation. Good morning, good afternoon, good evening, depending on the time zone you are in, and welcome to Energen's webinar series. My name is Jana Waldman. I'm the marketing director here in Energen, and I'll be your host for today. We are hoping you're safe and healthy, and thank you for joining us today. Today, we'd like to show you Energen's new customized genotyping solution called SNPer. We will explain to you the logic behind how SNPer works and how you can optimize and lower the cost of your breeding program, no matter what organism you are working on. We will save time for questions at the end, and we encourage you to type in yours during the presentation using the question tab on your screen. This webinar is also recorded and will be posted on our website in case you want to listen to it once again. I'm very happy to present today's speaker. Dr. Sharon Rehav is Energen's Senior Director of Genomic Solutions, and he is leading the team of our technical experts. Sharon has been working in the ag biotech industry for over 10 years. Being a third generation of farmers, Sharon began his education with agronomy studies in the Hebrew University of Jerusalem and continued it with a PhD in molecular genetics and system biology from the Weizmann Institute of Science. And with that, I'm going to turn over to Sharon, and he will start his presentation.
Thank you, Jana. We have a lot to share with you today. A brief caption of what's going to happen is I'm going to give a very brief introduction into Energen in case some of our audience has not been in contact with us in the past. A very small introduction to genotyping, what is it good for, why do we want it, and then SNPer, the topic of today's talk, and I'm going to showcase a specific use case with real data that we've recently done, establishing a small SNP panel for cannabis and hemp genotyping. So a little bit about Energene. Energene is a big data company. We've been around for around 10 years. We have a little short of 50 employees. Most of them, the R&D and headquarters are in Israel, and we have subsidiaries in the US and in Canada. We have a bunch of customers. We've been published quite a lot recently, and in the right side you can see a few of our strategic customers. Our vision is to enhance agriculture performance for our customers to achieve nutritional, the increase in nutritional value and securing food supply or agriculture supply in general. So a little bit about what we are actually talking about. We're talking about the application of molecular breeding and the return of investment on molecular breeding is explained in this slide. Basically, traditional breeding relies on crossing and selection. To make the right selection, you usually spend a lot of time and effort growing and maintaining a population of a certain size so that you can actually test the performance and select the product. Limiting factor here is the amount of resources and time that are spent, and they dictate to a large degree the size of the population that you are maintaining, and the size also dictates the likelihood of success, of reaching a successful product. In molecular breeding, what you are actually doing is you are virtually increasing by up to a few folds or even more the size of the population that you are screening by applying some sort of selection that's based on DNA test at an earlier stage. And therefore, you are actually maintaining and phenotyping a much smaller population, but it is highly enriched with individuals that are likely to have the attributes, the traits that you are after. So this is true for molecular breeding in all colors and shades, and I'm going to now show a few of those colors. Obviously, it's not fully comprehensive, but it's a way to actually view the different applications, and we've sorted them on the x-axis. As you see, the marker density that is required per sample, and on the y-axis we'll try to give some estimate as to the actual cost per sample to genotype. Just a word, if somebody doesn't know, genotyping means tests that are done nowadays at the DNA level, at the DNA sequence level, that distinguish between individuals of the same species, so that you can apply different levels of selection. So at the very low densities of markers per sample, you can find marker-assisted selection. This is essentially you have a trait that's predicted by a single marker. You have a marker per trait, or you have different QC type of analysis. This is not directly related to breeding, but it is important for anyone that that has a big pipeline of different biological samples to maintain. It usually doesn't involve a lot of markers that are being put to use. Starting to increase in marker-assisted backcross, you need genome-wide representation of a low-density set of SNPs so that you can apply selection to reach fully integrated individual within the shortest amount of time, or the fewer generations between introgression and achieving a new line. Genomic selection is actually a machine learning type of analysis that is being put to use in the recent years very successfully, in which you need to have moderate to high marker coverage across the genome and phenotype data on a training population, and that training population allows you to predict the phenotype of additional samples using the genotype only. And trait discovery requires usually a high density of information per sample. It has many uses to find QTL so that you can develop single trait markers, or to identify genes that you can turn into genome editing targets, or other applications that I would not go into right now. So these different densities translate into different costs, and those different costs are also displayed by different genotyping platforms that can be roughly divided into 4 different classes. There's the single marker assays. There's a lot of technologies here. I won't go into all of them. They can be very cheap when applied on a mass scale. Amplicon sequencing is an evolving technology that usually is applied to medium density of a few hundreds to a few thousands of DNA tests per sample of markers. Arrays are being put to use aggressively in breeding for a long time. They usually are best equipped to handle a very large number of samples, and also a relatively high density of markers. And genotype by sequencing can be very flexible, covering different densities for different applications, but there's only there's a fixed price per sample that's usually assigned to it, that you cannot really go lower than that. So these are the different genotyping platforms. I can talk more about it, and there's a good possibility that people that are now listening know more about those platforms than I do. But basically, this is the world where everyone that's doing breeding are operating, and the main motivation of SNPer is to try to make sense of it and try to fit the right panel, the right set of SNPs, and the right application for a specific aim. So I gave you a preview of what's written here. What is SNPer? It's a fully customized, all-inclusive genotyping solution that utilizes Energene's both the experience and the available data that we have for different species. It features a few different items. First is sequence-based diversity analysis, so that you can design the right solution for a specific population or project. That sequence-based diversity analysis culminates into a customized SNP panel that you can use. And also something to bring to the table is to select the right genotyping platform and provider that can give you the best results for that specific panel. Another thing that Energene promotes is the wide use of imputation to drive cost down, meaning you do not have to measure each and every data point that is needed on each and every sample. You can substitute a lot of the empirical measurements with a computational process that Energene can provide. And it should be cheaper than the empirical measurement when applied correctly. So those are the advantages. First, and I would say very important, is better quality. We strongly advocate that you invest once in designing an optimal marker set that is best fitted for the diversity that you are using. That initial investment allows you to actually reduce the cost by measuring less data points. Using a smaller SNP panel results in lowering of genotyping costs, and it can be further lowered if imputation is applied, as I suggested before. Another advantage is that it's highly flexible. The platform is designed to be compatible with any species and any application. and we also maintain flexibility in selecting the right genotyping platform and provider, and essentially you don't have to commit to any specific platform. If next year someone will come with a new technology that offers some advantages, you can basically switch to that. And updates in the SNP panel and other types of updates are available and accounted for. This is how it actually looks like. So there are two main stages in SNPer. The first is, as I said before, diversity analysis. You select a few founder lines that represent the diversity that you are working on. Usually you select a few more so that other parameters can be displayed, like allele frequencies and others. You then detect a very high resolution description of the diversity, and based on that you can design an optimal SNP set. This is all done by Energy. And after you have that, you can go into routine use. In the routine use, you can either measure the same set that was offered in the diversity analysis, or you can even apply a different strategy by measuring a subset or using a subset of that set on the bulk majority of samples and imputing up to that higher density that you've reached. And as I said before, updates can also be applied as time goes by, as diversity changes, and other things that might occur. In this presentation, in this webinar, I'm going to focus on the first part. I'm going to showcase how Energene has discovered and screened variants or SNPs for a specific application. The application is a generic small classification set of markers that can be used for exploring diversity in cannabis and hemp. I would speak about how we actually produce such a set and the applications of that to the cannabis and hemp industry would be further review in a dedicated webinar that's scheduled for next month, I think. So let's talk about the designing the SNP set that you are using. There's a very simple plot here. On the x-axis, you have the number of selected SNPs, and on the y-axis, you have the information content. The type of curve that you expect to get is a saturation one, meaning as you go from 1 SNP to 2 SNPs and to 100 SNPs, the incremental value in information content of each and every SNP is high, and as you approach the tens of thousands of variants, the addition of an extra one would not add that much information. In this space, everyone that's doing breeding needs to find the sweet spot of where do you exactly want your genotyping to be. There are other axes here, like for example, the price for a given number of selected SNPs, or maybe the price for different densities, but this is just to actually show you the main logic behind it. So let's say that we've done that on a specific population and we have that curve. We can also plot the information content and the density of an existing solution. For example, an off-the-shelf high-density array that people are using. That is very likely not to be that informative as a custom designed SNP, since there's a good chance many of the markers that are included are not informative in a given population. In any case, if that point falls on the curve, then that basically means that you're doing fine and maybe you shouldn't change anything at all. Our assumption is that there is room for improvement for most SNP sets. If you do find such a case, you can improve it in different ways. You can say, I have a model that works very well for me. So I don't need to increase the information content. I just want to go here in parallel to the x-axis and reduce the cost. That's one way to go about it. Another one says, I'm very comfortable with my current price per sample, but I realize that I can use the same density of markers, but increase the information content. That would increase my accuracy and result in better breeding. Another way to go about it is actually to do both, to both increase the accuracy and reduce the cost, and find a way here in between to actually establish the SNP panel that you are after. So from now on, I'm going to start diving into real data. So the real example that we are examining is the design of a small SNP set for cannabis and hemp. This specific use case has some shared parameters with, for example, designing a SNP panel for genomic selection, but there's also a couple of differences. The reason why we are showcasing this and not another is the fact that this is data that is available for us to share, while data that comes from commercial customers that are doing breeding, we are not at liberty to share. So the panel purpose that we've set out is to design a small SNP panel that can be used for any cannabis and hemp sample, and can be used for fingerprinting, QA, true to type, rough genetic classification. Those are all different ways of describing. I want to be able to differentiate between different samples that I'm working on, or possibly comparing them to varieties that exist elsewhere for different applications. And also to have a good measurement of the relatedness between different samples by comparing how much of the diversity between them is shared. So the input material that we had is short read sequencing of 46 diverse samples. I would speak a bit more about those samples as we go on. And the analysis main stages are as following. First, you need to produce a variant panel. I would show you a bit about the complexities of doing that and the way that Energene has solved it in this case. After you have a variant panel, you need to filter SNPs, and the filtering of SNPs needs to create a subset, a very small one, that is both clean, reliable, robust, informative, and compatible with the genotyping platform of choice. You also need to make sure that the SNPs are well distributed across the genome, and then you need to calibrate and validate them on actual data. I'm going to cover the computational side of things, as we do not want to dwell too much into cannabis and hemp biology in this instance. So the first stage is to produce a variant panel. So what people usually do is they take resequence data. 30x coverage is kind of a golden rule that a lot of people are using, although sometimes you don't need as much, and sometimes, at least for a few samples, you need much more. But in any case, you can align those reads to a reference and then conduct variant calling. The problem in this case, and actually in many other cases of plant genomics, is that there are shortcomings to this approach. One is the quality of reference that you are using. Second is the level of diversity that you are trying to capture, because aligning reads in cannabis and actually in other plant species may only account for 50% or even less of the reads mapping in high reliability to a single reference. And that's why we felt it's needed to apply a different pipeline that is, I would say, more or less unique to Energene. We can take those same reads and we can apply de novo assembly on those reads and assemble them into phased contigs or scaffolds. This allows us to both filter the sequencing noise or errors, basically reaching a high, extremely high reliability of sequences. Those contigs are also phased, meaning each contigs represent a single phase that is actually found in a specific sample. And as it's longer, it holds more information and can be aligned with high reliability to the reference. After we have done that, we've also mapped those scaffolds onto the reference and applied variant calling in a proprietary computational pipeline that Energene has developed. These two pipelines are we run them in parallel and for each sample individually. Each one suggests a few tens of millions of variants per sample in this case. You can then try and merge them. In this case, as we are aiming for a highly reliable set of SNPs that is much smaller than the variant panel that's constructed, we've applied a conservative approach where we actually wanted to find only the variants that are confirmed by both pipelines, meaning a highly reliable set of variants that you can use. After we've done that, we have a variant panel, and we need only to perform recalling of those variants on all of the lines together. So we again take the same input data and the reference panel or the variant panel that was constructed, resulting in a comprehensive, high quality genotype data for all samples, meaning a variant calling on each sample, on each of the variants that were discovered by this pipeline. Those are the results of that pipeline when applied to those 46 cannabis and hemp samples. So you have here a column for each sample. For each sample, the majority of SNPs were like the reference, but there was a considerable amount, roughly 5 million variants that distinguished that sample from the reference. And when taking all together, this is the total number of variants that we could find. So it's a little over 21 million variants. 86% of them are SNPs. Around 11% of them are short indels, and around 3% are not biallelic, meaning triallelic or more. So this is our starting point. This is the variant panel that we've constructed, and we now need to skim it down in a way that results in a SNP set that we can actually put to use in genotyping. And this is the next step, filtering of SNPs. So we have that initial number of 21 million, and it's a very large number. So the first thing that we did is to skim it down significantly by finding the cleanest subset of it. By clean, I mean we concentrated only on SNPs because most genotyping platforms operate best on SNPs. We also, in this case, chose to keep only variants that were localized to chromosome and not to unmapped scaffolds or contigs. And very strict criteria that was used here is we want them to be very distant from other variants. So the distribution of variants across the genome is not homogeneous. There are places where you can find many polymorphisms and places where there are fewer. And to apply genotyping successfully, you really need to find the subset of SNPs that are relatively devoid of other SNPs in their proximity. So that's the first filter. Second filter is reliability. We wanted to make sure that all of the SNPs that we are using are true. Although we applied many stringent criteria before, there's still a chance that some of the variants that we discovered are artifacts of some sorts. So we screened for the most reliable ones based on different, there are actually several quality criteria that were applied here. The second thing that is very important in this case and may be different in other cases is you wanted to make sure that those SNPs are robust. By robust, I mean that we can, as far as we know from the 46 samples that we have, we can be certain that for any sample, we would be able to call these SNPs, meaning it would not be in an area in the genome that, for example, is absent from some of the individuals. So we applied very strict criteria here. The next filter is around informative, meaning we wanted to make sure that the allele frequency, the minor allele frequency, is not small, meaning it's relatively large, so that in the most case, most SNPs would be informative when comparing to different samples. In other applications, I have to say that those two filters, the robustness and the informative, would maybe be switched. For example, in a given breeding population, the informativeness and the non-redundancy of SNPs, meaning are they linked in the same haplotype or are they orthogonal to one another, may be more important and would be placed with higher priority in this funnel. The last parameter that is actually kind of plug and play is platform compatible. So all genotyping platforms have different quality criteria for them to successfully produce a readout of a certain genotype on a given set of SNPs. All of them usually rely on the same parameters. You need to look at GC content and at the number of ends, at the repetitiveness of the region of the SNPs and other parameters. In this case, we've used parameters that are best equipped for amplicon sequencing. And the third, so before I go into that, so we've actually did this funnel starting with 21 million SNPs and resulting with around 12,000 SNPs that are reliable, robust, informative, and compatible. But you need to screen them a bit further. And the next criteria is to actually pick the one that are best spread across the genome. So in order to do that, you first need to bin the genome. So it's very simple. If you are, like we have done in this case, if you want to reach the ultimate goal of having a set of 200 SNPs, you need to divide the genome into 200 different bins. Ideally, that would be done on a genetic map, meaning we would assess what SNPs should be added from which region based on the likelihood of recombination between them. When a genetic map is not available, like in this case, you can use a physical one, and you can basically bin the genome into a specific size. In this case, for 200 bins, you need around 4 million bases in each bin. That resulted in a bit more than 200 because you can't actually concatenate two adjacent chromosomes. That does not make sense. So we had a few extra bins in this case when dividing an 850 million bases genome into 200 or so portions. What we see here below is actually the distribution of the different steps in the funnel across the 200 bins, just to make sure that we have managed to put enough candidate SNPs at each bin. So you can see here this grayish type represent the 30K, the robust SNPs. The lighter blue is the set of informative, and the darker blue is the one that are also compatible. That's the 12,000 SNPs that we've used. And just to make sure that we have enough, we can look at the distribution of the number of SNPs per bean, and you can actually see that the smallest amount per bean that was found was 9, and you need to select only one from them, so that still gives you a lot of choice to maneuver, and at the maximum you have 300 different SNPs to choose from. So it's a good distribution across the genome. It does not, if you look here, does not have a lot of bias as to too many SNPs in one region or the other. So we are good to go for the next stage. Next stage is actually to make the final decision. And that final decision is based not on a filter, but on actually ranking of the SNPs per bean according to a few criteria. The first and most important is informativity. To the left, you see the distribution of the minor allele frequency of the SNPs, and you can see that all of them qualify a certain criteria of being above 0.1, meaning above the minor allele frequency is more than 10% of the 46 samples. But you can still apply some more stringent thresholds and be as close to 0.5 as possible. And there are other criteria in place. One is you can still, again, filter the SNPs for their likelihood of being true. And also you can prefer ones that are positions at the center of the bean rather than at its edges, because then that would put them as distant from one another as possible, and that would increase their or reduce the chances of them being linked. As for any big data analysis, you want to allow some random selection so that you don't bias your selection too much on things that perhaps are correlated with the ranking that you have applied. So after we've done this ranking, we've basically divided those SNPs into 3 quality categories, A, B, and C, and those are the distributions of the different categories per bean. And using that, we were able to, this is the genome-wide distribution of those filters. So you can actually see how A, B, and C are located, and those are actually the final selection that we could make. So if you want for N1, meaning to choose one SNP per bean across the genome, almost all of the SNPs that were chosen were of class A, meaning they all had the maximal score using our ranking system, and only a few had class B. And if you want to choose 3, there would be a bit more class B, but still their ranking would be relatively high, and that could enable, for example, doing a larger panel if needed. So up until now, we've actually got to a candidate SNP set of around 200 SNPs. Now we need to validate how well does it perform discriminating different samples from one another, and this is the next few tests that we've applied. We first applied the principal component analysis on different set of SNPs in the process. One is using all of the reliable SNPs. So here you see only, I'm sorry, I'm not giving you the full detail, as I'm already presenting a lot of information. So we are only viewing the principal component 1 and 2 overlaid, and the different colors are actually represent the sources of the different samples. And you can actually see that, well, that's to be expected, that we can differentiate those different samples from one another quite easily. You can actually start to see, for example, that these 3 are very much alike and are actually triplicate of very similar biological material. When you reduce it to the 12K that we presented, the discrimination is actually better, meaning some of the some of the reduction that we did actually improved the result. You can actually see that the components are explaining more of the observed diversity than before. And you actually see those different sources of material clustering well together. Now, the actual question is, can we reach that same clustering when applied to 200 SNPs? And the answer is basically yes. You can actually see them very well distributed. And from that perspective, we are, I think, good to go. And after viewing that, we've continued to exploring other things that are more, I would say, down to earth, more easy to understand. The first, these are the actual genotype results of all 205 chosen SNPs on all 46 samples. And those scores were used to cluster them into clades using Euclidic distances between the scores. So you can actually see that for each sample, for each column, there's a distinct pattern here, except for these few cases. This is a case where you actually see two samples that are near identical, and here you see three samples that are very much alike. Those are results that we were actually expecting, having known a few things about those samples. Another interesting thing about those two samples is that they belong to a clade of samples that are also those are all the orange ones here, that are very much different from the other samples and are more similar to one another. This is the things that you would expect when looking at different breeding material, and we've actually seen that in many species, that these types of genome-wide analysis confirm things that people know to be true by pedigree information and other types of information. So let's ask, how well does this 200 SNPs really differentiate between the different samples? So this is a distribution of the number of divergent alleles, meaning if, for example, two samples have one allele difference between them, it's counted as one. If one is homozygote like the reference and the other is homozygote like the alternate, there's two alleles that are different. And we've done all the different, a little more than 1,000 pairwise comparison within that set of 46 samples. And we could actually see that the mean or median difference between any two samples is around 160, 170, meaning 40% of the alleles between them are different. And obviously the minimum is this almost identical biological repeat. This is actually a good representation of the error rate that you expect to make. So maybe a single mistake out of the whole panel. And the maximal distance is actually very large. And you can differentiate between the ones that we know to be sort of repeats between the main bulk of the samples that can really be distinguished by a large proportion of the SNPs that were chosen. So that's, I think, the final test that we've done to make sure that the whole process, starting from raw sequence data through 21 million variants and ending with 200 SNPs that need to encompass in the best way how to differentiate between two samples of these species is actually checked and verified. So to sum up, before we take some questions, we highly recommend everyone to invest upfront in understanding your germplasm. This is important both to optimize and adjust your genotyping to reduce the cost, but it also has additional benefits that I didn't get into, but that upfront investment can result in many other activities that your breeding may benefit from. Diversity analysis of complex and diverse genomes requires experience and competence. I hope that I've used this use case in cannabis to show you that Energene has such experience and competence, and we would be happy to talk with you about how you can leverage our experience to better your business. As I said throughout the talk, we have two other webinars coming. One would actually talk about the same SNP panel that I've just presented, and not how it was presented, how it was developed, but actually what it is good for and how it can be applied specifically for cannabis and hemp. And the other would focus mainly on the application of imputation as ways to make your breeding process, namely genomic selection in soybean, more efficient and more cost effective, so that you can genotype more samples and increase your gains. So those two webinars are coming up, and with that said, I would be happy to answer any questions you may have.
Thank you, Sharon. We still have a little bit time for the Q&A session, and we have our first question here. And the question is, when can we use SNPer in any breeding program? What are the prerequisites?
Okay, thank you for that question. So I would say that the best time is when you already have, for example, in the application of genomic selection, when you already have your training population, you already have phenotype data, and you are on your way to actually applying genotyping at a large scale. Up until that stage, you may have been able to afford using higher density information on that population, because there weren't a lot of samples at that stage. But when you want to really upscale, you would benefit from the application of SNPer. But obviously there are other use cases. SNPer can also be used to design smaller panels for marker-assisted backcross and other applications. So I was just referring to one.
And then the next one will be, how could we extend this data for the lines which are not sequenced?
Okay, that's a very good question. Thank you. So we would want to probe preferably founder material, meaning lines that that have been used repeatedly when you have throughout your breeding, and that are likely to hold the majority of haplotypes that are segregating in all of the others. If we sequence those, we are very likely to have most of the haplotypes represented. And then I would recommend also sequencing, perhaps to a lesser coverage, a wider pool, just to assess the allele frequency and linkage disequilibrium in your population. But I would be happy to talk to whoever asked that question and see exactly how many and what available data there is, so that we can find the optimal solution here.
So the next question is, does number of SNPs required based on genome size? In big genomes like chili hot pepper, will 232 SNPs are enough?
Okay, so genome size is important in the initial stage because you want to have sufficient coverage. For chili pepper, the genome size is 3.5 giga, so you would 30x of that would be much more sequence data than, for example, a cucurbit like melon or cucumber. By the way, there are other bigger genomes than pepper. When you come to the actual genotyping, the physical size of the genome has lesser importance than the actual genetic size of the genome. So the maize genome is smaller than pepper, but the maize genetic map has around 2000 centimorgans. So if you want a centimorgan resolution coverage of that, you would need 2000 SNPs. It may be that in pepper or in any other organism, the recombination frequency could be lower, and that could stretch up and down the number of SNPs that you would want. Eventually, at any given genome size, we hope to reduce it to a certain number of SNPs that do the job. And when that is actually applied, the original physical size of the genome becomes of lesser relevance.
The next question is, does it work for aneuploidy?
Again, an excellent question. So at Energene, we consider allopolyploidy to be the same as diploid, meaning if you have a polyploid species like wheat or like cotton that is an allotetraploid, there's enough difference between the two subgenomes so that we can find the subset of SNPs that are unique to a single homolog set. If you are talking about an autopolyploid like potato, for example, then it gets a bit trickier. And then you would probably need to make use of SNPs that at least some of them do not distinguish between the subgenomes, and that can be done, but would maybe affect for the worse the prediction accuracies that you can expect to get. Hope that answered the question.
And the next one is, would this be able to find the best plants for creating the F1 generation that has hybrid vigor?
Okay, that's a question about predictive genomics. When applied, genomic selection can be applied both to predict the additive effect within a certain heterotic group or specific pool. There's another way that can be applied when you want to predict combining ability or hybrid vigor. In any case, you would need to genotype, probably by the same set, and you would need a training population and a test population. The experimental designs of these are a bit different, but the genotyping side is I think, fairly similar.
The next one is, how many founder lines are recommended to construct a screening panel?
Well, that depends on the level of diversity that is explored. Some breeding programs are focused on relatively low diversity, so you can basically detect most of the segregating haplotypes by only sequencing a few. And some use higher diversity and are also introgressing more material as they go. We need to have a good grasp of what's actually known and what's unknown. And we can also apply an incremental approach where you start with a few and then you assess, have you covered enough, and maybe add a few more. And also, I have to say, there's other aspects that involve here, like budget, meaning how what is your ability, or how much is it worth to expand the diversity that you are exploring, viewing your business. So all of this can be accounted for. I don't have a magic number of, I don't know, 10 or 20 or another number of founders that would work in any case.
So the next question is, can I add pre-selected SNPs or gene of interest linked SNPs in panel?
Yes, I think this is highly recommended, that if you are already doing a SNP panel that you want to routinely use, if you can add your trait markers or markers for regions of interest for other reasons, that would allow you to actually use that set for different application. For example, for both genomic selection and for marker-assisted selection. When we recommend on a genotyping platform, we try to recommend the most flexible ones that can both utilize those extra SNPs and can also be very much adaptable to adding additional ones as the program matures. So the short answer is yes, it's possible.
Okay, I think it will be our last question as the time goes by. So the last will be, can the snipper be used in background selection? How much SNPs can be used in case of rice background selection?
Yeah, so for background selection, you don't need a whole lot of SNPs. But as I showed, a snipper can be applied to get to any density that you're after. I think when speaking more broadly about snipper, do we want to apply it also for the the all-inclusive genotyping? Is whether the application through Energene can meet other demands of background selection, which are usually very fast turnaround time. And that are aspects that we are very much interested in exploring, and we are actually diving into those issues with different customers, and we would very much like to discuss that with you and see if there's a way to actually do it together. The first part of the diversity analysis can be applied for any density, so that's kind of a given.
Thank you so much, Sharon. I'm sorry we couldn't answer all your questions. There are plenty great questions, but I can assure you that we will follow up by email to answer all of them. Give us a few days and we definitely will get back to all of you. I'd like to thank again Sharon for his time today and the great presentation. This webinar was recorded and we'll have it available in the next few days, so keep an eye for our email with a link to the recording. You'll be also able to find it on our website. If you have further questions or an interesting research project and like to discuss it with our experts, please don't hesitate to contact us at energene.com. Thank you for joining us today. Stay healthy, stay safe, and enjoy the rest of your day.
Thank you.